We often think about animal recognition in terms of, well, sight or smell.
Right.
Bright color, maybe a specific scent.
Yeah, the obvious cues.
Exactly. But what if it came down to something you could feel? Like literally the physical touch.
It's amazing how often nature works on these less obvious channels, isn't it? Especially on the micro scale.
Totally. And it's where we're heading today into the really quite amazing world of Caenorhabditis elegans, these tiny little worms.
C. elegans, yeah. A classic model organism.
Right. And we're not talking big displays here. We're diving deep into how something like body stiffness, how firm the worm is, actually helps males pick out a mate.
It sounds almost bizarre, but the research is pretty compelling. Our main source today is a paper called Body Stiffness is a Mechanical Property that Facilitates Contact-Mediated Mate Recognition in Caenorhabditis elegans.
Oh.
And it lays out this really neat two-step process that the males use. It's all about physical cues.
A two-step process. Interesting. So our mission for this deep dive is to unpack that. How did they do it? You mentioned two key things the researchers found.
Yeah, that's right. First, there's an initial check based on cues on the worm's surface, the cuticle, sort of a first impression. But then, for the male to actually, stick around and pursue mating, the body stiffness of the potential partner becomes crucial.
Okay, let's break that down. Step one, that initial contact. What's going on there? What kind of cues we're talking about?
So, when a male bumps into another worm, it uses these really short-range cues. It's basically touch.
Right.
And through that touch, it figures out the species, the sex, and importantly, if the other worm is reproductively mature.
How did the researchers figure that out?
Well, they use something called a male retention assay. It's quite clever, actually.
Okay.
You have a male worm on a patch of food. It's happy place, basically. Then you introduce another worm. If the male finds it suitable, a potential mate, it tends to just stay put on the food patch with it.
Ah, so it stops exploring and decides this is worth staying for.
Exactly. And they saw males would consistently stay with adult hermaphrodites.
Hermaphrodites, right, the main reproductive form in C. elegans.
Correct. But if they put in other males, or even different nematode species like Steinernema or Pristiontis, the male would just keep wandering, exploring.
So a clear discrimination happening right away.
Definitely. And it seemed like the discrimination was strongest at the genus level. They could easily tell, you're not even close to my species.
That makes sense. And what about maturity? Does that
Okay.
matter for this initial check?
Oh, absolutely. Males showed a strong preference for adult hermaphrodites compared to larvae, or the dower stage worms.
Dowers being that stress-resistant stage.
Yeah, the non-feeding alternative larval stage, so clearly not ready for mating. Interestingly, though, once a hermaphrodite was an adult, its specific age didn't seem to matter much for this initial retention.
Okay, so adult is the key signal there. Where are these signals coming from on the worm?
The evidence points strongly to the hypodermis. That's the outermost layer of living cells, sort of like their skin under the cuticle. They did some really neat genetic tricks. They could make a hermaphrodite's hypodermis more male-like.
Now what happened?
It became much less attractive to the males. They didn't want to stay.
Wow.
And conversely, if they made a male's tail hypodermis more female-like, its attractiveness actually went up.
That's incredible. So it's really about the signals coming from that specific tissue layer.
It really seems so. And they back this up by looking at genes involved in hypodermal development genes like Lin4, Lin28, BLUMP1.
Right.
Mutating these affected attractiveness. For instance, disrupting Lin4 leads to a more immature hypodermis, and those worms were less attractive. Messing with Lin28 or BLUMP1 also reduced attractiveness. It all points to the maturity state of the hypodermis being broadcast somehow.
You know, intuitively, I might have guessed the very outermost layer, the cuticle itself, the non-living part would be the main thing, or maybe chemicals on it.
That's a reasonable thought. But the researchers tested that. They tried washing the worms with organic solvents, looked at mutants with known changes in their surface coat. Surprisingly little effect on male retention. It suggests that initial attraction, that stay or go decision, isn't primarily driven by the chemistry of that very outer surface coat.
Huh. So if it's not the coat, but related to the cuticle somehow, what is happening at that first touch?
This is interesting. They managed to purify just the cuticles, like the shed skins of the worms. And when they presented these purified hermaphrodite cuticles to males, the males showed a brief transient response, They'd back up, turn around, a quick reaction.
So they do sense something on the cuticle itself.
Yes, but it's fleeting. Importantly, this happened even with cuticles from GP7 mutants. These are worms we'll talk more about. They have stiffness defects.
Right.
And this brief response was species-specific and stage-specific. So the cuticle does seem to carry some basic, I am an adult, C. elegans info for that initial contact.
Okay, so it's like a quick ID check from the cuticle. But you said it was transient, short-lived.
Exactly. It suggests these cuticular cues are for that very first assessment. But remember how manipulating the sexual identity of the hypodermis affected retention, but didn't seem to change this brief cuticle response?
Oh, right.
That hints that figuring out the sex for a sustained interaction, that crucial piece, probably involves more than just those initial cuticle cues, which brings us beautifully to the second step.
Body stiffness.
Okay.
I'm ready. This sounds really counterintuitive. How did they connect mate recognition to, well, how squishy the worm is?
It started with a genetic screen. They were looking for genes that, when mutated, made hermaphrodites unattractive to males.
Okay.
And they hit upon this group of genes encoding what they called furrow collagens. Genes like DREPI2, DREPI3, DREPI7, DREPI10.
Droopy often means dumpy, right? Shorter, fatter worms.
Often, yes.
Yeah.
And mutations in these specific furrow collagen genes caused a dramatic drop in attractiveness. Males just weren't interested in staying with these mutant hermaphrodites.
So these collagens must be important. Furrow collagens. I guess they're involved in making those ridges or furrows on the worm's cuticle.
That's exactly right. These collagens help form those circumferential wrinkles. But what was really telling was that other mutants that affected body length or shape, like GP5 or LON2, didn't necessarily lose their attractiveness.
Uh-huh. So it wasn't just about being short or dumpy. It was something specific to these furrow collagen mutants.
Precisely. It pointed away from just general size or shape and towards something related to the function of these specific collagens.
So maybe the furrows themselves are the key signal, like the males are feeling the texture.
That was a hypothesis. They focused a lot on GP7 because it had such a strong effect. They confirmed its role is in the hypodermis, that skin layer again.
Mm-hmm.
And interestingly, this rule seems conserved in related Caenorhabditis species. Get this. If they made male worms overproduce DPY7 protein, those males became more attractive to other males.
That's wild. Really underscores DPY7's role in broadcasting some kind of mate quality signal, even if it's misplaced there.
Exactly. It's presenting mate information. And they found more evidence using temperature-sensitive versions of DPY7.
How did that help?
Well, at different temperatures, these mutants showed different effects on body shape versus male retention. It suggested DPY7 might have separate jobs, one in overall structure, and another specific to this mate recognition signal.
So maybe it's not just the physical presence of the furrows.
Seems that way. Because they found another mutant, DREPI18, which was unattractive but actually still had detectable furrows.
Okay, that complicates the feeling the furrows idea.
Right. And using high-powered microscopy, SEM, they confirmed that mutants like DREPI2, DREPI7, DREPI10 lacked clear furrows, while others like DREPI5 had them. But attractiveness didn't perfectly track with just having furrows or not.
So, if it's not just the furrows, what else do these furrow collagens affect?
This is the key link. Body stiffness? They measured it directly using atomic force microscopy, AFM. Basically, poking the worms very precisely. And the furrow collagen mutants that were unattractive, they were also significantly less stiff. Floppier, essentially.
Wow. Okay, so the lack of these specific collagens makes the worms less stiff, and that's what males don't like.
That became the strong hypothesis, so they tested it more directly.
How?
Well, one way was just like gently poking worms to let out some internal pressure, made them less stiff.
And did that affect attractiveness?
Yep. Low male retention, just like the fur mutants. They also looked at a different mutant, GON2, known independently to have reduced body stiffness. Same result, less attractive.
Okay, the link looks pretty solid.
Even how they prepared the worms mattered. Fixation methods that made the worms softer also reduced male retention. It all pointed towards stiffness being sensed.
It really does sound like the males are kind of assessing their firmness, a tactile check.
You could definitely frame it that way, but crucially, stiffness alone isn't enough.
Okay. Why not?
Remember Pristinchus pacificus, that other nematode? It turns out its stiffness is actually pretty similar to C. elegans.
But C. elegans males aren't interested in it.
Right. And we already know purified cuticles alone don't sustain interest either.
So it really needs both parts, that two-step mechanism you mentioned at the start.
Exactly. Step one, Initial contact cues on the cuticle saying, roughly, I'm the right species, right stage. Step 2. Appropriate body stiffness for sustained interest, maybe signaling health or quality.
And did they test that two-step idea directly?
They did with a really elegant experiment. They took those P. pacificus worms normally ignored, right?
Right.
And they coated them with purified cuticles from D-Pi7 mutant C. elegans. Remember, these cuticles can trigger the initial brief contact response, even though the worm they came from isn't stiff enough.
Okay, so P. pacificus body plus C. elegans initial contact signal.
Exactly. And guess what? It worked. The C. elegans males showed the initial contact response, and male retention significantly increased.
Wow. So you need the hello signal from the cuticle, even a faulty one in this case, combined with a physical body of roughly the right stiffness.
Precisely. It strongly supports the two-step model. You need both components.
That's a super clever experiment. What about the other extreme? Can a worm be too stiff?
Good question. They explored that too, using tiny 3D-printed bionic worms.
Seriously? Bionic worms?
Yeah, made of a stiff resin. On their own, males weren't interested at all.
Okay.
Then they coated these very stiff bionic worms with the DREP7 mutant cuticles. They got the initial contact response.
But did the males stay?
Nope. Negligible effect on retention.
So it's not just stiff is good. There's a sweet spot.
It really suggests there's an optimal range of stiffness they're looking for. Not too floppy, not too rigid.
The Goldilocks zone of worm firmness. That makes sense biologically.
It does. And to bring it all back home, they checked if this actually matters for, making baby worms.
Right. Does it affect reproductive success?
Absolutely. They compared mating success. The DREP7 mutants, the less stiff, less attractive ones, had significantly fewer offspring from mating compared to normal worms, or even compared to other dumpy mutants like DREP13 that were still attractive.
So this whole elaborate touch-based system directly impacts their ability to reproduce effectively.
Without a doubt. It shows how vital this contact-mediated recognition is for their fitness.
Okay, let's try and sum this up. It's pretty amazing. These tiny worms have this incredibly sophisticated way to find a mate, all based on touch.
Yeah, it's a two-stage process.
First, that initial contact using cues on the cuticle, kind of a quick, are you C. elegans, are you mature?
Yeah.
Heck.
The initial handshake, basically.
And then, crucially, step 2 is assessing the body stiffness, which seems linked to these furrow collagens. That confirms suitability and leads to sustained interaction.
Exactly. It's a mechanical property check that follows the initial chemical or structural cue check.
It really expands how we think about sensory input, doesn't it?
Yeah.
Moving beyond just smell and sight to include these mechanosensory cues in social behavior.
Absolutely. It highlights the importance of mechanics in biology, even in behavior.
And of course, like all good research, it leaves us with more questions.
Always. Like, what exactly are those initial cuticular cues? Which molecules?
Yeah, and how precisely do the males sense stiffness? What are the neurons, the receptors involved in feeling that difference?
And the neural circuits, right? How does the brain process stiff enough versus too floppy and translate that into a decision to mate or move on? Lots to figure out still.
Definitely. It really makes you pause and think, though. How many other animal interactions, things we think are simple, might involve these hidden layers of sensory information, these tactile conversations we're just starting to eavesdrop on?
It's a great point. Perhaps touch and mechanics play a much bigger role across the animal kingdom than we currently appreciate. If you found this interesting, definitely check out the original paper. It's fascinating work. And the whole field of C. elegans behavior and mechanosensation is full of surprises.